Okay, as promised in the last lecture, I said that I will put a video cast together for you to demonstrate the principles of docking a drug into a biomolecular target. And what I'm going to choose as an example to illustrate a point is the HIV protease 1OHR.pdb. So this is X-ray data from an experimental database, and that particular protein has a drug bound to it, but I want to show you how the principles of docking a drug to a target are going to work using this as an example. So what we'll do is to open Mo. I first import the PDB. So this is the raw X-ray data from the crystallographic database, and here we have it. What I want to show you first, just so to teach you a little bit about the enzyme, is that I've just coloured the cartoon for the protein with two colours. The protein HIV protease is actually made up of two units that are coded for by the virus that make a dimer that is only active as the dimer, and we're going to do various things with this set of data. First of all, what I would like to do is to remove the inhibitor. What we're actually going to do is to reintroduce the inhibitor back into the binding site to show you how faithful the docking is, but the principles would be applied to developing new chemical entities to design new chemical entities to fit into this binding site to inhibit the protein, the active enzyme. So this graphic is showing only part of the active. Is showing only part of the protein structure. So the computer package actually knows to show you relevant amino acids that are local to the inhibitor. The first thing we've got to do is actually prepare the protein for modelling, because as you can see, hopefully as you can see here, if I make this a little bigger, you can see that there's some missing data from the protein. So it knows, because of the nature of the format of the file, it knows that there's some missing coordinates from the X-ray data. Well, we can correct these elements, and you'll see the various missing excuse me, the various missing atoms of the various amino acids that have low X-ray resolution within the experiment are corrected. Their positions are calculated and built back into the model. So that puts some amino acids back into the structure. We're then going to protonate the structure, which allows the titratable protons to move between acidic sites and basic sites to approximate how the protein would be at physiological pHs. So if I move this out of the way, hopefully, if you can see a recalculation in progress, you may just see protons moving around, but you don't, because this is actually, in this case you don't, because this is quite a small protein. And that's done very quickly. Okay, so now our structure is ready for modelling. Just to check that, if I calculate the potential energy, here is the total potential energy of the system, stretching, angle, I'm not sure what that is, outer plane, torsions and so on. Hold on. van der Waals interactions, electrostatic interactions. So it's got all of the parameters now within that system for molecular modelling, and that's what it needs to be able to dock and assess the energy of binding of an inhibitor molecule. So our molecule is now ready for action. We're going to look for the active site of the molecule, and we have a system called Site Finder, which works within this molecular modelling package and allows us to locate areas of interest. So if I now apply Site Finder, we get this area just above this pocket, and hopefully you can see that there is a sort of a cavity there. The way Site Finder works is it places, if I select all the sites it's found, you will notice there's actually various other sites on the protein, but actually the one of interest is the top site, and that is ranked in order of likelihood of binding, of being able to bind a drug molecule. Now what you'll notice is white and red spheres. The white spheres are likely positions in which a hydrophobic atom would be found, and the red spheres are the likelihood of a position where a hydrophilic atom could be found. And just to show you that how this works, I'm going to attempt to show you now. If I choose a lipophilic surface map coding of the molecule, and try and take the surface away so you can maybe see what's happening a little bit. So what we have here is a map of the hydrophobic and hydrophilic regions of the protein, calculated by the surface properties of the amino acids within the molecule. And I don't know how clearly you can see it, but let's choose this region here. You can see that this region is coded purple on the map, and you notice that there's a collection of red spheres. Well, the purple coding is actually a hydrophilic region within the protein, and then other regions, if I can find an appropriate region, maybe you can't see it so well here, but let's try. So here, where you have a collection of white spheres, you will see that there's a green region surrounding these white spheres. So this is green here, all through here, and you can see that there's some green actually there's a green patch here next to these white spheres. So showing a map of surface properties hopefully allows you to think about how these spheres are located in the molecule. Now the spheres are placed randomly all through the surface of the molecule, and where those spheres energetically bind favourably, the spheres are retained. It's a bit like shaking sticky white spheres and sticky red spheres all through the put the protein molecule in a box of sticky white spheres and sticky red spheres, shake them about, put them randomly in positions within a box with your proteins sitting in them, and where they stick at the most likely sites is probably going to be the site where your drug will want to bind. Okay, so now that we've done that, there are a couple of things, technical things you have to do. What we're going to do now is we're going to hide the centers, and you'll notice they've gone. We're going to set dummies atoms at this site, which the dummy atoms are required for a docking process that's going to occur in a moment. And what you notice is that there are smaller spheres in that cavity. Okay, now what we need is our drug molecule. So what I'm going to do now is bring in a drug molecule, and the drug molecule is ready for docking. I'm just going to hide it so we don't complicate what's going on. And now we start a docking routine, and the program is known as dock. We have to set up various things here. So the site is the receptor atoms, the receptor is the receptor atoms, the site is the dummy atoms that are displayed. In this particular instance, I'm not going to rotate bonds. That's going to make the job a little easier and quicker so you can see it in real time. And there are various technical ways of matching your structure to the white and red spheres, and scoring with respect to energy the outcome. I'll show that to you in a moment. But first of all, what we're going to do is get the job running. And we've got a database file already existing. We've got a blank table at the moment, which is going to populate with information at a later stage, and I'll show that to you in a moment. But hopefully what you can see now is a docking in process. Now, the way that the docking works is it looks at the molecule, the inhibitor of interest, and it tries to match by a statistical fitting procedure that occurs in 3 dimensions the hydrophobic atoms and the hydrophilic atoms, or the atom typing, with the dummy spheres that we've put in, the red and white spheres, so that the pharmacophoric elements of that ligand will be binding to the binding to the amino acids of the pockets that is being presented in the most appropriate way. And you can see that there are various poses. So the attempt to fit the molecule is known as a pose. There are various poses which allow the molecule to fit to these white and red spheres. And then when the pose has been settled, a force field it comes into play, which fits the inhibitor against the receptor, optimizing bond lengths, bond angles, torsion angles, the electrostatic interactions, the van der Waals interactions. And as part of that process, it also scores the ligand with respect to the ability to bind to that to this site that we're looking at. I'll show you some figures in a moment on the populated table as to what's happening. You'll notice as the things update, as the system is updating, information is appearing graphically as well. And what we're seeing here are interactions between hydrogen bond donors and acceptors as possibilities, interactions between atoms in the center of rings. There's lots of things that the detection program picks up as elements of interest for the molecular modeler. Now it takes a little while to do this. What I'm going to do is I'm going to reintroduce the surface map that we saw earlier, but only restrict it to sorry about that, to near the dummy atoms. It's going to be a molecular surface of the receptor atoms. I'm going to code it with respect to lipophilicity. It's going to recalculate the surface and display it in the area of interest. So hopefully that you'll see what surface mapping. Oh, sorry. Let me near the dummy atoms. Let me redo that while the docking's still taking place. Hopefully it'll give us a modified map of the properties of the pocket. Yeah, here we are. So hopefully we can see the docking in progress, and we can see properties of the pocket that the drug is trying to fit into. So you'll notice that here is our, if I tip it this way, our pink region here in the pocket contains the red spheres. A green region here contains predominantly white spheres, which means a hydrophobic part of the drug will bind there. And if you actually notice on the, if I zoom in here, this drug is being docked at the moment in an interactive way, and you can see that it's actually trying to map a cyclohexyl type ring to that particular collection of white spheres. So it obviously is determining that the drug actually likes that position. But it doesn't only do the cyclohexyl. It poses another part of the molecule, trying to sort out whether that molecule, a different functional group on that molecule, wants to bind there as well as an alternate pose. Now some of those will be successful and some won't be, and some poses will be rejected and others will be accepted as the system is scored in terms of energy. And you notice that another pose here is trying to place our cyclohexyl ring. So mapping the properties of a pocket can allow us, sorry if I just reset the view, mapping the properties of a pocket can allow us to analyze features that a drug molecule or an inhibitor might want to see. What I'm going to do is I'm going to recode the surface with a different property, and in this case it's electrostatics, and you see a white, red and a blue coding, and the red coding will be a negative region attracting positive charges. The white region will be hopefully coinciding with white regions of the map where electrostatic properties are not necessarily important. And then a blue region will be a region where so here we have a sphere in the blue region. That's an area where that will most likely attract a negative charge on our ligand. So what I'm going to do now, the docking is actually finished, and here is a table that's been produced of our 5 structures that are our top likely structures, and there are various figures to be looked at here, but the figure that is used is this S value, and you can see it's a negative energy, which means it's an attractive interaction. It's in kilocalories per mole, and there's a range of energies, and our position 1 is the top docked position because it's the most negative or most favorable position. Now what I'm going to do is I'm going to browse through the possible conformations. There's only 5. This is number 1, and what we can do is we can study the various possible poses that are kept, and so what I'm going to do is retain structure 1, and I'm going to colour the atoms of that structure, let's say yellow. Now what I'm going to do is show you exactly where the ligand binds. Now I'm going to hide, so you can see what's going on. I'm going to hide the protein. I'm going to hide the surface map, and here you have a solution to where that drug molecule wants to bind in the active site. The red structure, if I hide the hydrogens on the structure so you can see them a little bit clearer, the red structure is actually where Viricept binds to the HIV protease, and the yellow structure is the solution to the docking for Viricept, treating it as, when I read the molecular file in for the inhibitor, it was actually a molecule of Viricept, and what it shows you is that it finds faithfully the docking position of Viricept within the HIV protease. Now hopefully that has shown you how docking can work for us.